All right, so we're on now to our third section of our chapter on genetics, chapter 12 in the Raven book, and we're starting off this section by talking about dihybrid crosses. So, like I mentioned in the previous section, a dihybrid cross is where we're going to look at two traits as they're tracked through the generations at the same time. So, the traits that we're going to look at in this case are going to be for seed texture and for seed color in garden peas. So just as kind of like a quick orientation here, the big R is going to be for round seeds, little r is going to be for wrinkled seeds, big Y is for yellow, little y is for green. All right, so for this first parent, we have round yellow, true breeding, and the second parent is wrinkled and green, true breeding. All right, so we're going to take those, and I mean, we could do a Punnett square, and like I said before, Punnett square for a dihybrid cross should have 16 boxes, but we actually don't need to do that for this one because it's really, really straightforward. All right, so the thing that we have to remember here is that these two genotypes, big R, big Y, and little r, little y, little y, both of those two genotypes are for a diploid cell. So if this diploid cell were to make haploid gametes, all of those gametes would have to have a big R and a big Y. There's no other option because this individual only has big R's and big Y's. So we know it's diploid because it has two of each. We know that if we're making gametes, the gametes have to have one of each. And so the only option for the gametes from this parent is to be big R, big Y. That's it. That's all it can be. No other possible option there. For the other parent, all of its gametes have to be little r, little y. Again, no other option there. That's the way it has to be for that parent. So if we take those big R, big Y gametes and cross those with a little r, little y gametes, which again are the only gametes we can possibly have here, then every single individual in the F1 generation has to be big R, big Y, just like this. So still round and yellow, but no longer true breeding. We have the big R and big Y from this parent, and little r and little y from the other parent up here, and so that gives us the genotype of the F1 generation. Now, the F2 generation is going to be a little more complicated because now there are going to be more possible gametes that the next generation can produce. So if we take this pair of genotypes, we're allowing the heterozygous individuals to cross with themselves, we are eventually going to see a 9 to 3 to 3 to 1 ratio. So in other words, 9/16 of the offspring are going to have round yellow, both dominant traits. 3/16 are going to have round green, 3/16 are going to have wrinkled yellow, and 1/16 is going to be wrinkled and green. And I'm going to prove that to you in a second by showing you a Punnett square that is for this particular trait. The thing to remember about this, when we do this cross here, big R, big Y, little y, times the same thing again, the thing that we have to remember is that this individual can only make so many different kinds of gametes, and every single one of those gametes must have one and only one R and one and only one Y. If the gamete has two R's or two Y's or no R's or no Y's, that's not going to work. That's not going to be a viable gamete, so it wouldn't actually produce a zygote, which is even viable in that case. So this individual here, again, if it's diploid, has two R's and two Y's, every single one of its gametes has to have one and only one R and one and only one Y. So there are four possible combinations. A quarter of the gametes are going to come out with big R, big Y. A quarter of them are going to come out with big R, little y. A quarter of them are going to have little r, big Y. And a quarter of them are going to be little r, little y. And that's what we have lined up across the bottom down here. This genotype parent can produce four different kinds of gametes. Big R, big Y, big R, little y, little r, big Y, little r, little y. And if both parents have this genotype and both parents are producing these gametes, then the outcome that we get is this, right? These combinations here across the top and along the sides are the gametes from the two parents, just like on that previous slide. Remember, big R, big Y, big R, little y, little r, big Y, little r, little y. Same thing here from parent 1, same thing here from parent 2. All right, so if this gamete, we'll say this is the male parent over here, and this is the female across the top. So if this sperm cell fertilizes that egg cell, then the zygote will be big R, big Y, little y. Look at this first box. If this sperm cell fertilizes that egg cell, then we're going to have big R, big Y, little y, which is what this box is saying. And if this sperm cell fertilizes that egg cell, then we're going to have big R, big Y, which is what that box is saying. So from there, we're just filling in boxes. But the thing that people really struggle with on doing Punnett squares, especially for a dihybrid cross, is setting up the sides and the top of the Punnett square. So what you need to remember is the sides and the top of the Punnett square represent the gametes of the two parents. The gametes are the combinations of letters that we find in the parent's genotype. Right? And again, these gametes can only have one and only one of each kind of letter for each gene that we see. So if we do that cross, where both parents are heterozygous for both traits, and we fill in all these boxes, if we add them up, we're going to find 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 of them are going to be yellow. We're going to have 4 of them that are green, but we need to go in more depth there and look at how many of them are round and yellow. If that's the case, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9/16 are round and yellow. If we count up the ones that are round and green, we have 1, 2, 3 that are round and green. How many are wrinkled in yellow? 1, 2, 3. So again, 3/16 are wrinkled in yellow. And then this last one down here is wrinkled in green. That's the 1/16 that remains. The genotype there is little r, little y, which is recessive for both traits. And so that's our Punnett square. Now again, not every parent is going to have this exact genotype, and therefore not every parent is going to produce those gametes, and therefore not every Punnett square is going to look like this across the sides and across the top. So what you need to understand is why these letters filled in here are what they are. That goes back to this, all right? The genotype of the parent and the gametes they produce are based on having one and only one of each kind of letter, R's, Y's, or whatever other letters that you happen to choose. So looking at what's happening here again with gamete formation, and this really goes to the heart of setting up a Punnett square, we have the focal individual here. This particular species has 6 chromosomes, so 3 purple, 3 blue, 6 total. That's the diploid condition. Now, what they actually have are 3 chromosomes from the dad and 3 chromosomes from the mom. And so 6 chromosomes total, which they can then partition up into different gametes randomly because of the principle of independent assortment during metaphase 1 of meiosis. There are 8 different possible gametes that this individual can produce. And you see why we're not doing this for humans, because if it were humans, there would be 2 to the 23rd power number of different possible gametes, which would be ridiculous, not even possible really, to sort it out that way on paper. So we keep it small with hypothetical species that have 2 or 3 or 4 or 5 or 6 chromosomes. Well, not 3 or 5, but 2, 4, or 6 chromosomes, and we really don't want to go beyond that. But again, the idea here is that when we're producing gametes, sometimes the gametes might just have dad's chromosomes. Sometimes they might have just mom's chromosomes, but more often than not, they're actually going to have some combination of mom's and dad's chromosomes that are sort of put together at random when the individual is producing their gametes during meiosis. All right, so that, again, is a throwback to Chapter 11 and our meiosis independent assortment discussion that we had back then. All right, now, what we just did, the dihybrid cross that we just did, we had an unspoken assumption that we were dealing with. And the assumption that we were dealing with there that I didn't talk about was the fact that both of these two genes are on different chromosomes. So for example, if we're dealing with a hypothetical species that only has 4 chromosomes, right? So maybe the, so 8 chromosomes total, right? 4 from mom, 4 from dad. Maybe the color gene was on chromosome number 1, and the texture gene was on chromosome number 3 or something, right? So the genes are on different chromosomes. So therefore, they're going to be inherited separately. On the other hand, what you can have sometimes is when you're looking at 2 different genes that happen to be on the same chromosome. And if that's the case, if 2 genes are on the same chromosome, they are going to tend to be inherited together. We're not going to get as complicated as this, because in that case, the genes tend to go together unless they happen to be separated during crossing over. So for the most part, when we're talking about genetics in this course, we're dealing with genes on different chromosomes because that's going to make things, honestly, a little bit easier to deal with at that point. All right, but just bear in mind that because each chromosome has hundreds or thousands of genes on it in real life, it's very possible at some point in time as a geneticist, they might want to track 2 different genes that actually are on the same chromosome, and at that point we would say they're linked. All right, now these 2 slides we're going to skip, and we're going to move on from there and talk about a test cross. Right? So a test cross is something which, pretty honestly is not really done anymore. But back in the old days, this actually used to be a really common, really useful technique for figuring out the genotype of an individual whose phenotype you knew. So, this sounds all well and good to say that a species has a big A or a little a or whatever. But remember, if you're looking at an animal or a plant or whatever it is it doesn't have letters written on it that you can then just read off and plug into a Punnett square. It has physical traits. It has a color. It has a coat type. It has a seed color. It has a physical trait that we're then trying to figure out what the genotype maybe is. So when that becomes particularly difficult is if we're dealing with a species that has a dominant trait. Because remember, a dominant trait could result from the individual being either homozygous dominant or being heterozygous. Either way, they would get the dominant trait showing up as part of the phenotype. So maybe we're dealing with a breeding program for livestock and we're really concerned about a harmful recessive allele being in the livestock that we don't want to pass on. We might then want to think about doing a test cross. So again, if we have an individual bull, say, that has a dominant trait, and the dominant trait is what we want, if we want to ensure that male is not carrying the little a, if we want to make sure that little a doesn't sneak into the offspring of that bull, what we would do is a test cross with that bull to try to figure out what his genotype is, and therefore whether or not he might be a good candidate for our breeding program. So, again, what we're dealing with here is trying to figure out whether the dominant phenotype is a result of a homozygous dominant genotype or from a heterozygous genotype. In that case, in the explanation I just gave or the scenario I just gave you, we would want to use the bull if he was homozygous dominant, but if he was heterozygous, we maybe want to find a different candidate for the breeding program and send that first guy off to the dog food factory or whatever. I don't know. I guess it wasn't nice, but we're not going to use him for the breeding program anyway because he has that little letter in there, that recessive letter in there that we don't want to pass on to the offspring. All right. So again, the basic idea here, with a dominant phenotype, you have an unknown genotype. You've got a big P, but you don't necessarily know what that second letter is. It could be a big P or it could be a little p, and you would still get the same phenotype. So what we would then do is cross that unknown individual with someone whose genotype we know, and that would be somebody who has the homozygous recessive condition. Because if they have the homozygous recessive condition, they're going to have a certain trait, and if we see that trait, then we automatically know what their genotype was. So the idea is this. We're going to take these two parents. We're going to take the unknown genotype parent and the known genotype parent, and we're going to cross them together, and we're going to see what the offspring look like. So if in this test cross, we're dealing with pea plants and flower color again, if this parent is homozygous dominant, then if we cross that parent with a homozygous recessive individual, then 100% of the offspring are going to come back as being heterozygous. So if this is the unknown parent's genotype, this is the known parent's genotype, all of the offspring should come back as being heterozygous. On the other hand, if this is the unknown parent's genotype, and this is the known parent's genotype is little p, then we should get on average half and half, dominant traits and recessive traits showing up as the phenotype. In reality, if we see even one recessive phenotype individual, we know that unknown parent had to have the recessive allele because there's no other way to get two little letters together, unless the one parent is big little and the second parent is little little. There's no other way to get two little p's in this case together. So, like I kind of mentioned to you before, most human traits are not quite as simple as what I've been showing you thus far in this lecture, right? Most human traits are controlled by more than one gene. So far, the traits we've been looking at are controlled by one and only one gene. Another assumption that we've been dealing with that I didn't really talk about so much yet was the fact that each gene that we've looked at so far only had two possible versions. For flower color, it was either purple or white. There was no other option. In reality, a lot of genetic inheritance is going to have more than two different alleles that are being tracked throughout the population during given crosses. We also assume that there was a simple, clear-cut, dominant recessive relationship between the two alleles in the population. That is also not always the case. There are cases where we will see things called codominance and incomplete dominance, where you actually end up having a bit of a sort of a hybrid phenotype in the offspring, but not because of blending inheritance. It's actually because of a incomplete dominance or codominant type situation. In reality, when Mendel published his work back in the 1860s, it really wasn't read by very many people because it was written in a journal that nobody really read. It was a fairly obscure journal, and it wasn't really translated into an English or a language, any other language that people read very often, so it was more or less undiscovered for about 30 to 40 years until it was eventually kind of found by some people who put Mendel's work together with Darwin's work and created what's called a modern synthesis of biology, which is basically it describes everything. When you look at Mendel, when you look at Darwin, when you put those ideas together, it is a really, really strong set of ideas about how species change over time and why species change over time. But this is, again, one of those cases where Mendel was long gone before people realized how important his work really was. Kind of the same thing with Rosalind Franklin we talked about back when we covered DNA. People didn't realize how important her work really was until after she'd passed away, which is really unfortunate. So one situation that is a bit of a complication or complicating factor when we look at how traits are inherited in a population is something called polygenic inheritance. So polygenic inheritance is a situation where more than one gene controls the phenotype. We don't just have one gene controlling one trait, maybe we have 3 genes controlling one trait. And so a lot of times whenever we have polygenic inheritance, we will see a range of phenotypes that are produced. We're not going to see just either purple or white. We're going to see 10 different colors that are the result of the combination of 3 different alleles in the population that you're inheriting from 2 different parents. So it gets a lot more complicated, but it is still not terribly complicated as compared to some traits. So a really good example of a polygenic trait is human height, right? So if we go back to pea plants for a second, pea plants come really in only 2 heights, at least genetically. Soil conditions and water and sunlight will affect their height. But as far as genetics, there are only 2 heights for pea plants. There is tall and there is short. That's it, right? There's literally 2 heights that you can get to, assuming all other conditions are the same if you're a pea plant. Now, humans clearly don't work that way. It's not the case where humans, everybody in the population is either 5'2" or 6'3", and there's nothing in between or on either side of those 2 ranges or numbers. There's a huge range of variation. In fact, there's an infinite range of variation between the shortest human on Earth and the tallest human on Earth. If you could measure it precisely enough, you would find an infinite range of variability within those 2 extreme ends of the size continuum for humans. So human height is actually not controlled by a single gene. It's controlled by a bunch of genes, a whole collection of genes that, along with interactions from the environment, like your mom's nutrition and her overall health when she was pregnant with you and those kinds of things, all of these things, your diet as a child, the nutrition you had, the medical care you had, the exercise you got when you were a kid, all of these things control your height, and there are a huge range of different possible heights in humans. So if we map that out, if we sort of take a population of, say, 100 humans and line them up based on height, so we have, not even very precise here, 5 foot, 5'1", 5'2", 5'3", all the way up to 6'5". If we look at the population as a whole, we actually get a bell curve. And that is very, very typical of traits that are controlled by polygenic inheritance. Is to get a bell-shaped curve where we have a few individuals at one extreme, a few individuals at the other extreme, and then a lot of individuals at some kind of more or less average value in the middle. That is a very typical situation for polygenic inheritance. Think back to pea plants for a second, where we only had 2 different alleles of one gene. For a graph of pea plant height, and again, assuming all other conditions are the same, you would have literally just 2 bars. You would have like this bar here. So we'll say it's this bar and this bar for pea plants. You'd have one set that were short, one set that were tall, and nothing else in between, right? That's not the case for humans. We have a bell-shaped curve as opposed to just 2 kind of different bars in the curve like you see with pea plants. Another good example of polygenic inheritance is human skin tone. Now, this can be a little confusing because this is not the same thing as albino or not. That's actually a different gene, which is a simple, clear-cut, dominant recessive relationship where you either make melanin or you don't. That's one gene with 2 different alleles, much like the inheritance we saw so far with flower color in pea plants or seed color in pea plants. But there are 3 other genes. Then we get 3 of those genes from our mom, 3 of those genes from our dad. There are 3 other genes that determine how much melanin you make, assuming you're able to make melanin. So assuming you're not albino, the combination of these 3 other genes will determine how much melanin you make and therefore how dark your skin is. So if you inherit 3 copies of the light allele from dad and 3 copies of the light allele from mom, then you're going to have all light alleles and you're going to have very, very light skin. At the other extreme, if you inherit all 3 dark alleles from dad and all 3 dark alleles from mom, you're going to have all dark alleles and they're going to have very dark colored skin. But there aren't just 2 skin tones in the population of humans. There's a huge range of variability from the lightest person on Earth, again, not albino, but just naturally doesn't make very much melanin. They make some, but not very much, to the darkest person on Earth, which makes a ton of melanin in their skin and hair and eyes and so forth. There's a lot of variation in between there, because we're dealing with more than just one gene. We're dealing with 3 different genes, 2 alleles, one from each parent for all 3 genes, which gives us a lot more variability when it comes to the offspring. All right, so what we're going to do now, we're going to cut off the next section. We will start up section 4 of a recorded lecture by looking at pleiotropy, which is a kind of a similar situation to polygenic, but in a very different way. And we'll start off with that and then continue on, and we're pretty close to finishing out this chapter, but not quite there yet.